Welcome. Thank you. There you go.
Okay. Thank you, Patrick, and thank you to the Dassault team for inviting me here. It's been a wonderful meeting. Today, I'm going to talk about atlases, which are collections of maps, and what it means to go where there are no maps, into uncharted territory, both in the literal sense and the metaphorical sense in cell biology. So in 1831, a young naturalist named Charles Darwin, who liked to collect beetles, he boarded a naval vessel bound to do an expedition around the world, carrying with him, sort of his father's disappointment and a mind primed for revelation. Darwin's trip on the Beagle took 5 years. They thought it would be 2. That tells you a bit about challenges in project planning, even in the 19th century. And it changed our view of nature and humanity's place in the world. In Galapagos, on the island chain off of the coast of Ecuador, he encountered finches, birds whose beaks seemed sculptured in different ways for different purposes. Each one on a different island had a different shaped beak. And he realized that this was due to evolution by natural selection. That's where his great discovery came from. And he figured that out, changed the world for the rest of us, but he didn't know the mechanism. He didn't know how traits changed and how they got passed from parent to child. That remained uncharted for another century and a half. And speaking about uncharted, last year I had a chance to retrace a small part of Darwin's voyage. I volunteered to be a crew member for a leg on a sailboat that was reproducing the voyage of the Beagle. And I joined it for just this leg right here through Polynesia. From Mangareva to Tahiti, we sailed 1,200 miles. And what you can see is that the captain avoided the Tuamotus. The original idea was to go through them, but the actual journey went around them. And that's because they're uncharted waters. Even in today's world, where we've learned so much about the Earth and around us, there's still uncharted waters, and just kind of incredible to me. So we're going to carry that idea forward now. The missing piece in Darwin's framework of evolution was the information storage and transmission mechanism. He had no idea how inheritance actually worked at the molecular level. We now know that it's about DNA and our genomes. DNA is a molecule, it's a digital storage mechanism like a molecular tape. It stores information using a 4-letter chemical alphabet. We have roughly 3 billion letters in our genome, and that encodes about 20,000 genes for all of us. And what Darwin couldn't see, and what we now know, is his entire journey was across an invisible map, the genetic map that connects all of life. Those finches, which were different species on different islands, shared 98% of their DNA with each other. But they also share a very large fraction with us, 50% with bananas, 25% with rice. Every stop on his journey, he was sampling a 4 billion-year-old tree of genetic relationships. Now, you might think this is done. Our journey's done. We've sequenced the human genome. So we've worked out our map, and the human organism is now charted. But it's not. It turns out the profound irony in all of this is solving the genome did not tell us the map of the human being. And there's very deep parts of that that we had yet to work out. So we don't know how the genome predicts the cell types. So humans have 20 trillion cells in our body, roughly speaking, thousands of different cell types. Each cell has the same genome in it, but assumes a different identity. A neuron, very different than a heart cell, very different than a lung cell. How does that work? That is sort of the journey I want to talk about today, working that out. And that's a question that has been around for a while. Sidney Brenner, the great biologist, 20 years ago, in his Nobel Prize lecture, pointed out that so genocentric has biology become, we've forgotten the real units of structure and function of the organism are the cell and not the genes. So he suggested back in 2002 that we go on this journey, trying to map all the cell types of the human body. And we managed to do it 20 years later, and I'm going to tell you a little bit now how we did it and what we've learned. So we have mapped out cell atlases of the mouse, of the fly, of the lemur, of the human. These were very large-scale projects. Papers had 150 authors. It was an amazing exercise in team science and a community effort, I helped found a new research institute to get these done, the Chan Zuckerberg BioHub. It was sort of an amazing journey, and I'll tell you a bit now how we did it and then what we learned. One of the key technologies used to do this at scale was microfluidic automation. These are the biological equivalent of the integrated circuit, little chips with automated plumbing on them, valves, pumps, and such forth, that was part of my group's research over a number of years. And these became the tools, these and their successors became the tools to automate single-cell genomics. And I just want to say, parenthetically, SolidWorks played a big role here. We used it not just to design the chips in the community, but also to design the devices they connected to. So thank you, Dassault, for that. It was amazing help in getting this all done. With this sort of high throughput automation, we were then able to do these projects with millions of cells. And let me give you a taste of one of the discoveries. So how do genes get controlled? Which genes get turned on in which cells and which not? It turns out some genes are controlled by other genes. This is known as the second secret of life. It was discovered by Jacob and Monod working in Paris. And in fact, you know, pretty much every picture of Monod, he's got a cigarette in his mouth. It's sort of very old school. The first secret of life was the structure of DNA. They discovered the second secret of life, which is how genes are regulated. And it turns out that there are proteins that will bind to DNA, attract the polymerase enzymes that transcribe the gene, and that's what turns genes on or off at any given moment in time. So the study of those genes is particularly important in understanding the definition of cell type. There are 1,622 of them that have been predicted to exist in the human genome, and we were able to study the expression of all of them. Every single one, for the first time, we've been able to identify its expression. And some of these are expressed, many of them, in just one or a handful of cell types. And you can see that in the figure here, where every column is a different transcription factor, every row is a different cell type, and the intensity of the color is the intensity of expression. So many of them expressed in just one or a few cell types. And this is how cell types are often defined, by the transcription factors that are expressed there. And Yamanaka showed 20 years ago that you can reprogram cell types using them, and it's very important to have this sort of map of transcription factor to cell type definition. Other transcription factors, shown on the right here, are expressed ubiquitously in all cell types. And these are sort of the foundational control things for cell division, metabolism, stress response, and such forth, properties that all cells share and are deep in evolutionary history of life. Among those, you'd think those have all been found by now. Turns out they haven't. We found many that had never been studied before. They were uncharacterized except for bioinformatic identification as potential transcription factors, and it's created sort of a wealth of new projects to understand about sort of universal cell behavior. Another thing we learned from the atlas was to study cells that were senescent. So senescent cells are associated with aging. They're the cells that have stopped dividing, and they're putting out inflammatory signals and other factors that sort of create trouble and lead to disrepair and dysregulation of tissues and organ systems. And they're the subject of a whole class of drugs called anti-senolytics meant to slow aging. And yet, when we were looking at our atlas, we discovered that we had the largest collection of human senescent cells ever studied before, 50,000 of them across the atlas. And so we began to ask the question, what defines senescent cells across 24 tissues in the human body? And we discovered there's not a single definition. It's not one gene expression program that defines all senescent cells. In fact, there's a broad class of them, maybe half a dozen. They use maybe a dozen or so gene expression programs. And we've been able to unravel some of that complexity to understand how different cell types age differently in different tissues. And you can imagine that's going to lead to a whole bunch of very interesting future practical work. So you might imagine there's practical applications that come from these cell atlases. And there are. I'm not going to dwell on this, but using the techniques we use to make the atlas, we and others have been trying to develop new drugs. These are two that I've personally been involved in. The first is a biologic drug. It's an antibody meant to cure food allergies, and we were able to find it by using the cell atlas measurement technologies to look at very rare cell types from the immune system of kids with food allergies, and discover those antibodies and create engineered versions of them that would be protective rather than harmful. And that is now in clinical trials in humans, and we'll know the results early next year, whether that's working or not. So that's an example in immunology. Another example in cancer, using small molecule therapeutics, is based on work that Mike Clark and I did for years, trying to use the cell atlas techniques to dissect tumor complexity and find new drug targets there. And sure enough, we found that KDM4 would be a very interesting target for small molecule drugs, and Jeff Stafford built a small molecule to drug that. That's in phase 1 trials right now, and we're very excited to see where that goes. And there's many others done by other people in the field. And so these atlases have led us to very important applications in human health. And developing a new drug is also a journey into uncharted territory. In the case of the allergy drug, it's a completely new method of action that nobody's ever tried before, so we'll see where that takes us. And in the case of KDM4, it's a first-in-class drug. No one's ever drugged that molecule, that protein before, that gene before, and we'll see what happens there. So let's return to Darwin, which we opened with, and the topic of evolution. So we spent a lot of time trying to think about how to interpret the data in the atlas. How do you assemble it into one coherent whole and have it be something other than just a list of cells and gene expression numbers? So we decided to use large language models to try to do that. And we would train a large language model on the atlas data to find an embedding space that would help us understand relationships between cell types. And we decided to be very ambitious and do this in a way that worked not just for one organism, but for all organisms. So we designed these models to be independent of the underlying genome of the organism, and decided to see what that could teach us about their value and about evolution. So this model that I'm describing here, developed with Theo Karaletzos at CZI, is called Transcriptformer. We trained it on 12 different species. It's a 500 million parameter large language model, transformer architecture and such forth, and represents sort of state-of-the-art in the application of these techniques to cell atlases. So with the fact that it could be used with different species, here's one application. It's very important, for example, in drug development, when you use model organisms to try to extrapolate from one organism to another, from rodent to primate to non-human primate to human. This is an example of using the Transcriptformer model to do that, looking at data generated by others who were studying innate immune response to LPS perturbation, which is like a simulation of a bacterial infection. And we looked at the ability of the model to use the data from one organism, like mouse, and predict it in the others, like rat, rabbit, or pig. And it was able to do that very well. And so that illustrates, the value of having a model that works on more than one species and why it might be important for some clinical applications. Here's another application. And that, of course, that data was not in the training set for the model. Here's another example of sort of out-of-distribution data. The model was trained entirely on healthy cells. We decided to look at published data on COVID-infected human lungs and see if the model could differentiate between cells that were infected with COVID and those that weren't. And it turns out the models did a very good job of that. And as we compared it to other models that, whoops, sorry, let's go back one, wrong way. As we compared it to other models developed that worked only on the human genome, the models with the architecture that worked for any genome performed better, even if we only trained it on human data. And so here's one with our model we trained only on human data, another on model organisms, another on all 12 metazoans. And so that's a very interesting discovery that with this out-of-distribution data, the structure of the model mattered a lot, independent of how you trained it. So you can also look at further out-of-distribution questions, like the ability of the model to classify cell types in species that the model had never seen before, never trained on. And this is ultimately where we want to go with this. We'd like to be able to use the model to predict cell types from the genome. That's the question I opened this with, you might recall, of a genome is never seen before, and to understand the cell types. Here, this is a small step towards that. We're using the actual measurements of species it's never seen before and asking it to classify cell types, and it's doing a pretty good job. It's a challenging question, it's out of distribution, and it gets more difficult the further back you go in time. So here, we have tested it on species that weren't part of the training, lemur, frog, lamprey, coral, and you can see the performance is starting to degrade as it goes back in time. So that's more work to do. I mean, that's sort of where we are at the state of the art. Interestingly, you could ask the question, do you want to train on more species? Would that help make the model better? Turns out not so much. The one we trained on 5 model organisms versus 12 metazoa performed comparably. So there's some other bit of cleverness we've got to work out here to solve this problem. Ultimately, I'm hoping that this is going to lead us to ask and answer other evolutionary questions. This is a graphical representation of cells from many different species embedded in the same space. One of the takeaways I want you to have here is having this genome-independent embedding space lets you measure distances between cell types of different species. It provides a mathematical way to measure distance between different cell types across species. We've known how to do that for individual genes for a long time. We now have the ability to do that at the cell type level. One of my pet questions has been to try to use this to understand the evolutionary origin of multicellularity. So we decided to embed single-cell organisms into that space and ask, what is the closest cell type from a multicellular organism to a single-cell organism like Baker's yeast? Turns out it's progenitor in stem cells, and that's providing a very interesting next avenue of investigation for us. So I'm going to wrap up here with sort of a philosophical comment about the value of models. And one that might be in opposition to the digital twin folks, so we can have a very sort of vigorous debate about that in the coffee hour. This is a short story written by Borges. This is the entire story. It's a 1-paragraph short story. I'll pause at the end so you can read it. But what it is, I'll summarize it briefly. It is the story of an empire of people who were very good at cartography, at making maps. Okay? And they were so good at making maps, they made maps that were perfect reproductions, right down to the scale of the city that it represented. And the following generations who weren't as fond found this useless, and the whole thing just turned into dust. The lesson is, the point of a model, in many cases, is not to be a perfect reproduction. And in the case of the cell atlas models, I always get asked, how come there's no proteins in there? How come we don't have dynamics? It's an abstraction. We think about the cell as a bag of RNA, and we're able to learn some very useful, important biology from that. And it's not meant to be a perfect reproduction. It's supposed to give us insight. And that, I think, is the point of the Borges story as well, about the value of maps as an abstraction. Thank you very much.
Thank you, Stephen. Transcript former. Now, that was impressive. I think we have time for questions, and I have like billions of them. So let's start with one. This large language model approach for lineage, if I understand correctly, is it pure generative AI learning, or are you guiding?
That's a great question. Okay, so it was very important to me that we not be sort of circular in how we did this. So I absolutely insisted that the model is trained only on the gene expression numbers. It was a matrix of numbers. Rows were the cells, columns were the genes, there was numbers in there. The model had no knowledge of biology. It had no knowledge of gene names. It only had numbers, not even gene names. It's just numbers.
And not even amino acids,
correct, none of that.
None of that.
Just literally a matrix of numbers. And the question then became, what biology emerged from that? And it turned out, and I didn't, for reasons of time, get into all this, that the model learned a lot of biology. The emergent behavior was fascinating. It learned a ton about lineage. I mean, the distances in the embedding space all have a lot of biological meaning, even though that wasn't part of the training. And that was why I'm so enthusiastic about these approaches,
so
actually.
It's almost evolution theory on steroid with LLMs a little bit.
Yeah, absolutely.
Another question that's very close to this one. In the future, do you want to guide the learning? I mean, how is modeling and simulation helping with the goal of the virtual cell and AI?
Yeah, so I think there is a point where we want to use our biological knowledge to help it as well. And I would say the place we're using that sort of most eagerly right now is in doing cell type annotation. So the way we did it originally, when we first made the atlases, is part of their value was that we did human annotation of every cell. It was the largest human annotated data set of this kind in the world, and it was very widely used for training algorithms and metrics because of the enormous amount of human effort and labor that went into annotating and creating a ground truth. But the flip side of that was it was a lot of work, and people began to get tired of doing that, and so we began to look at automated ways to do that. So we found some of the foundation models like Claude, and a paper is about to come out on that from our group, have been very good at doing automated annotation.
Okay.
And so you can give them the differential gene expression, let them use their knowledge of all of biology, and ask if these cells have certain genes differentially expressed, how would you label this cell? And they do a pretty good job at that now.
So specialized fine-tuning, if I understand.
Yeah. No, we're not fine-tuning the model.
Okay.
We're sitting on top of the model, but using its responses. It's more prompt engineering than fine-tuning.
Prompt engineering.
Yeah.
Okay. Any questions from the room here to Stephen? Over here?
Thank you for a great talk. I'm curious if you encountered any what I would assume would be called ontological surprises in the atlas when you looked at classifications and found things that were either surprised that went against what biology has traditionally told us, either from cell biology or biochemistry. Did you find anything interesting in that way?
Yeah, I mean, this is sort of why we're doing the yeast and the single cell stuff, is to try to understand that, because no one's had the ability to ask those questions before. And so to find that single-celled organisms, the eukaryotic precursors to multicellularity, are close to stem cells has got us scratching our heads. You know, I didn't talk about this, but one of the projects we've been interested in has been using this for understanding convergent evolution. And so that's where there's a lot of surprises. So convergent evolution is when nature has found the same solution twice. Birds and bats both have wings, but not a common ancestor with wings. One of the classic examples is the box camera eye, which humans have and octopuses have, but no common ancestor has. So we decided to look at that. We made an atlas of the octopus eye and looked at the human eye data and compared them together. And you know, one of the surprises there was that every cell type mapped very well. And so then it's like, could it be that all these cell types were evolved independently by octopus and humans? So then we began mapping to the fly eye. And it's a compound eye, different structure, and they all map just as well. So wait a minute, what's going on? So the conclusion is that all the cell types to make the eye, no matter what the structure, are ancestral. And that is something that I think nobody had anticipated before.
Interesting. Any other question? I have one, because it's so interesting. Looking at the embedded space, your embeddings, and you know that there's a trend which is more and more representation engineering to try to understand the chaos in those latent spaces. Did you observe patterns at a higher level than just genes to try to infer some logical structure between species or between cells?
I mean, that's a super interesting question. And there's sort of a technical answer. We're starting to scratch through that. I mean, I wouldn't say we've really unlocked it, but if you look at the transcriptomal preprint, there's a whole section about
that should be the next step,
yeah,
right?
it is. Exactly. It is, for sure.
All right, let's thank Stephen for those impressive works.
Thank you.
Transcript former.